Okay, so our role in this project is to kind of map what's going on in different variants, and we've been using, oops. Basically, the cell line that we've been using is the HEK293 cell line, which is a human cell line, and we use it because it's very amenable to genetic editing. So the base editors that we use to generate the cell lines, this cell line is easy to generate these mutations in. Most everything we've been doing, I put this up for the families, I won't go into it, has been done with the E420K cell line, which was actually very amenable. It was almost perfect for our BE4 base editing. We have 2 sites, and the idea is you have to target this single G in basically billions of base pairs. The base editors have a window, the best one we have, of basically 5. And so you hit the G, and not the adjacent A's. They won't affect that. And so this was an easy one to produce. The 198K has been giving us trouble in the respect that the editing window here has 2 Gs, and we have to hit this one without hitting this one, or vice versa. And so we've been generating cell lines that have both. And also the other challenge is we have to hit just one allele. And so we've got lots of cell lines that have double mutations and homozygous mutations for 2. And so we may have to make better tools for that. But the other technique that another approach that we're going down is to make the knockout cell line, which we have done. So we took the 293 cells and we used the classic CRISPR methods to just eliminate the entire R5D. And then we can put back the different variants into the cell line that doesn't have the others. And this was literally just made in the last couple weeks. Okay, so what I'm going to I'll show you today is that we've generated a much larger data set. Last time we talked about we had the heterozygous mutation for the E420K, and as Veer Lee mentioned, with some of the data sets, you can't tell if the phosphorylation change is on the wild type allele or in the variant allele. So Chinta developed another cell line, which is E420K homozygous, which means that the mutations occur in both alleles. And so we can use this as an additional control along with the heterozygous and the wild type to give us a better understanding of these pathways. And so we've got another data set with the phosphoproteomics, and then we also looked at changes in global proteomics, so changes in total protein levels. And then I'll give you some data on the updates of the pathway. Okay, so this is just for the scientists. The idea is to eliminate the R5D. We use classic CRISPR. So we just targeted exon 4, and we have one allele had a 5-base deletion, the other had a single base insertion. They both lead to premature stop codons, and that leads to nonsense-mediated decay. I put the data in. It's clean, so I'm not going to go through it, but this is just for everybody. And this is the take-home. So the take-home is basically, it was successful. This is the wild type. This was just grown plus or minus serum. And this is R5D. And in the knockout, we're not seeing any expression. So this actually may have some relevance to there's one variant. I remember it's the R85 star, in which case you have a stop codon. And what that leads to here is basically you get a message, and they're degraded if they don't have the proper tailing. And so what's probably happening in this variant is they're actually getting a suppression of expression. So they'll probably have 50% levels of the normal or some variation of that. Anyhow, so this cell line's made, and we're just getting ready to put that through the phosphoproteomics when we put the different variants back in. So the plan then is that the knockout cell line will be useful because we can look at changes. Remember that this is a holoenzyme where R5D is just one of the B prime family that are assembled into these trimers that function. And so we'll take this out and we can look to see if there's compensation for the other family members, if they go up or down, and then we can put the different variants in. And this we can share with everybody so everyone who has the variants can look at different aspects. The other thing that we've done is we've increased our phosphoproteomics capability. Our menu has a new mass spec, and it allows us to look at 12 samples at one time. So we've repeated what we did. This is what we reported last time, where we basically compared the wild type versus the E420K variant. And the idea is we simply take the cells, we get 4 or 5 biological replicates of each, you cut them up into peptides, you enrich for phosphopeptides, and then you heavy and light label them and mix them, and then you get mass spec analysis of everything at the same time. With the new machine, we can actually now do heavy, intermediate, and light labeling. So the idea here is we're cutting the cells with proteases, and then we use formaldehyde. If you use just C13, normal carbon, normal hydrogen, we have light labeling. We can use deuterium to intermediate label, and then we can add C13 and deuterium to give us heavy labeling. And this allows us to look at 3 different groups at the same time, all at this, basically doing the same MS run. So this gives us a lot more power in the analysis. And the data set literally just came in a couple days ago, and I got this from Chinta on Sunday, so it's really new, but it's striking. So we're looking at here, these are the volcano plots showing these are looking at proteomics, so these are changes in protein levels, and then phosphoproteomics. This is changing in phosphorylation in the peptides. And the blue are decreased, and the yellow are basically peptides, or in this case, peptides from proteins that are increased in abundance and then in hyperphosphorylation. And what's striking to me about this is how similar these plots look. And if you look, we have a protein-corrected plot. And for that, what we're talking about is these are changes in phosphopeptides when we've subtracted changes where the protein levels went up or down. And again, there's a lot of similarity. If you look, there's a lot of spots that are actually very close. And so the next step is to validate these. And this is what we were doing with the last data set, to look at the pathways. And the pathway that jumped out was this AKT-mTOR pathway. These are actually the spots that were hyperphosphorylated, leading to, this is serine 473 and AKT, and then these are phosphorylations that are inhibitory sites on GSK. And this has all been validated. We use a Western analysis for an orthogonal validation of everything that we see in the mass spec. So the idea is the mass spec identified these as hyperphosphorylated, and then we get the phosphospecific antibodies and compare. And so far, this has been matching up quite nicely. So the addition, and this is something that Chinta has done, so she's taken the homozygous cells, so these are the cells that have the variants in both alleles, and we're predicting that this is a dominant mutation, because that's what you see in the children where you have one allele mutated, and then you see the effect. And that looks like that's true. So far we're going back to validate the pathway with the homozygous cell line. So here's 3 replicates with the homozygous, 3 with the heterozygous, and the wild type. And again, this AKT-mTOR pathway that Rita talked about is being validated. We can add AKT2. We had AKT3 last time. We get AKT2 phosphorylation. And then this serine, this is an activating phosphorylation that now we're quite confident in. And the same thing, this is Gsk3. This is a downstream target of AKT. This phosphorylation is actually an inhibitory site on Gsk3 alpha and beta activity. And again, the homozygous, there's 3, the heterozygous. So this looks like we're getting further validation of this pathway. So the other thing I want to call attention to, and I brought this up at the May meeting, is that the changes are occurring at a level of regulation, at least what we look at. So if you look at S6 phosphorylation, which is downstream in the mTOR pathway, and if you look, these are the normal cells, the wild type cells. As the cells go from a low density culture to a high density culture, the cells make contact and they have pathways that signal density arrest. And so this is important in normal organ development where the cell's liver has to grow to a certain size and then it stops. And then in wounding, you have a cut, the cells will grow to fill in that cut and then they'll stop. And so S6 is a readout. This actually is when the cells are in log phase, they're not very confluent, and then the phosphorylation goes down. And the variant, the E420K, it's up all the time. So maybe it's a little easier here. So this is the normal. So as the cells become confluent, the S6 phosphorylation goes down. In the variant, we don't see that. So as they become confluent, this pathway remains elevated. It may not be as high as it could go, but it is certainly above where it would be in the normal density arrest. And you see the same thing when you look at growth factor signaling. So S6 phosphorylation is also affected by serum or growth factors. So this is the S6. So this is looking at the density. This is the wild type cells when they're pretty much confluent. If you take the serum away, S6 phosphorylation goes away almost completely. And then you can add back. EGF is a growth factor, IGF insulin-like growth factor 1. In the absence of serum, in the cells that are arrested, you can turn this pathway on. So this is another mechanism where you have metabolic or growth factor control that would normally turn this on even when the cells are, in this case, density arrested. And you see the same thing in both. This is the S6 and the AKT. In the variant, again, it's on all the time. This is the normal pathway. So this is the arrested cells. Take away the serum, S6 goes away. EGF, IGF, and the combination of the two, you get an increase in signal. In the variants, we're seeing this on all the time. It's not necessarily on at the maximal level, but it's not responding to the normal growth factor signals. So this is an interest, and the other interest that we have is going downstream. As mentioned earlier, rapamycin and the other TOR inhibitors are effective at suppressing this pathway. From the mass spec data, we see phosphorylation here. These are all downstream targets of TOR that we would expect, but we're also seeing things that are upstream. And when we treat the cells with rapamycin, these are the variants that have it on all the time. This is a dose-dependent suppression of, in this case, S6, which is downstream, but we're not seeing changes in the AKT or the GSK, which are upstream. So we wanted to further explore if we have something that can modulate this, what happens to these other pathways? So we want to explore what's going on here. And the two things that I have to share with you today are looking at Gsk3 signaling. And we're looking at, in this case, this is a transcription factor called MYC. And MYC is a major regulator. This is one of the first oncogenic mechanisms that was identified. MYC is a major regulator of cell proliferation. And there's a lot known about it. So in MYC, normally in response to growth factor signaling, you can get phosphorylation on a serine 62. And then as a suppression mechanism, Gsk3 beta can phosphorylate 3 58. And the idea here is that phosphorylation of MYC leads to the stabilization of this transcription factor, which is now inducing the expression of genes. And then dephosphorylation and degradation of MYC occurs through a two-step process where you have this phosphorylation followed by the removal of the serine 62, and then that leads to the degradation of MYC. And in both, here's the heterozygous and the homozygous versus the wild type, we're seeing a hyperphosphorylation of this serine 62. And this is actually consistent with the inhibition of GSK. So this is an inhibitory phosphorylation site. So you would see less of that. So you would see less phosphorylation and less degradation. And so we've got, with the MYC antibody itself, we actually have two bands. And again, this is the wild type and the homozygous. And we're not sure what this upper band is. If it's MYC, then we're having a change in the size. So there's something that's being physically changed. If it's just a nonspecific band, then we're seeing a change in the MYC protein levels as well. And so this is a new avenue to explore. And this actually fits a lot of the proteomic data. There are a lot of proteins that go up that are probably regulated by transcription. And the other is CREB, which was one of the first. CREB is the cyclic AMP responsive element. So basically, this is a transcription factor that binds to changes in metabolism that regulate cyclic AMP and then influences genes that control a lot of basic metabolism, glycogen metabolism. And we see this is a key phosphorylation site. So in CREB, phosphorylation at serine 133 is known to be associated with the activation of cyclic AMP signaling at an expression level. And again, an AFT1 is basically an ortholog. They're very similar. These are two proteins that are kind of redundant as transcription factors. And again, here this is the heterozygous 3 homozygous. We're seeing hyperphosphorylation of both CREF and AFT1. And so this basically means we need to go in and explore the changes in the mRNA levels. And we're positioned to do that as well. So we can do the RNA-seq with the heterozygous, homozygous, and wild-type cell lines. And so what this is bringing us back to is the mechanism of what's going on. And so the hypothesis that Stefan put up a while back was that pKa, phosphorylation and activation of this serine 573, was involved in the activation of to a holoenzyme causing a signaling cascade. We can add an AKT feedback loop here to the phosphorylation of CREB with the data, and we need to explore the possibility that one is involved in this, or maybe this is a more direct path with 2A. And so I guess the discussion that's come up, now we have the holoenzyme assembly issues. This is the model, and so there's this, if this is involved in the stability of the holoenzyme complex, if you have mutations, say here and here, that are causing that to basically dissociate at a slightly different level, and this might be, the E198 seems to be stronger, this may be weaker. And the other thing is that if this is a regulatory loop or it's fitting back in, then the phosphorylation, if this is a contact that's needed for inhibition, you mutate this, you have less of a regulation. You mutate this, it might be actually a more severe alteration in this negative domain. And so the crystal structure will be really nice if we can see how this really fits and how the contacts fit. And the other is the substrates. The substrates could fit in this groove. This is very close to the active site. And so if this is pushing the substrates in or preventing the substrates, the mutations can go in. So our plan then is to figure out more of this pathway, and all that proteomics data I will give to Carol to put in the Dropbox, and so that everyone. Yeah, this is actually a much bigger data set. It's a lot deeper, and there's literally about 50,000 proteins. Okay, and with that, I want to stop and thank all the people that are doing all the work. So Chinta is here, and she's been paramount in this effort. She's really been the star in the lab, getting the cell line and getting the westerns, and Mark has been really critical in helping design this and doing the base editing. And then we've added Alla. She's a great lab manager that will come in, and she helps with the tissue culture. Laura was a medical student that came in over the summer that actually worked on the knockout cell lines. And then I didn't talk about it today, but Chin Lee and Vito Richter is another graduate student. They've been working on the PDE another aspect we're looking at is there's a phosphodiesterase that changed, and we should be able to get some meaningful information out of that soon. And then Arminia Kettenbach, she's the mass spec doing the phosphoproteomics.
So Richard, you were talking, in one of your slides, you were talking about how rapamycin seems to have an impact downstream in inhibiting the mutation or the impacts, but nothing upstream. Does that suggest that, were you kind of suggesting maybe there's something else that would impact upstream, or?
Well, yeah, I mean, so that's what the data, so yeah, so if you look at this is the variant and the wild type. So the rapamycin, what's exciting about this is it's working at both. So that's putting this pathway under the control of the inhibitors. So if this is hyperphosphorylated in the variants by itself, so this is a mechanism in this cell line to regulate that.
But would that be considered normal regulation then?
Well, you're using a drug to regulate
but
it.
Would it stimulate normal regulation?
Well, it could be used to compensate for the abnormal regulation. So you're seeing this is a lot more phosphorylation than here, and so you can use the drug to hopefully take this back to this. But we don't see a change in, this is AKT, which is up here, and then GSK, which is down here. So one of our goals is to actually map out, there will probably be one or two, or maybe 10 at the way things are going, sites that R5D is working on as a substrate. I mean, so that there's going to be hopefully a protein we can say, okay, this is where it all starts. And in the meantime, we're just trying to figure out, and we're trying to do that simultaneously, but we figure out this pathway has become apparent, so what's downstream? And there are drugs that target GSK3. There are drugs that target PI3, and so what can we do to further regulate this pathway?
So does that suggest you might actually be two drugs?
Yeah, or more. I mean, yeah.
And this is all at a cell level we're talking.
This is all. This is cell, and it's one type of cell,
Yeah.
and it's a fairly messed up cell genetically. I mean, but we're using it because it's, well, for genetic engineering to manipulate the genome, it's very amenable. And there's also a lot known about the biology of the HEK293 cells. So there's a lot of literature. So we can say, this is what's normally happening, and we don't have to go back and rediscover it.